I would like to thank the organising committee for inviting me to present the 2022 Lowenthal Lecture. It is a privilege and honour for me to do so. I shall be talking on PET-based radiomics for personalised medicine. Many years ago, when I was a radiologist training in nuclear medicine, I commissioned this cartoon to sum up the relationship between the two specialties at that time. On the left, we have a nuclear physician concentrating all their attention on measuring the flow through the pipework in this system, unaware of the structure of the pipework on the other face. And here we have the radiologist concentrating very much on the pipework on this side of the face, but unaware of the flow through the pipework. And yet if we stand back and see both faces at once, we have a much clearer idea of the disease process. Now, many things have changed in the 30-odd years since then, and one of the first things to happen was that radiologists started to look around on the other side and measure flow, and I was one of them. Here we have a CT-derived image of renal blood flow that myself and colleagues from Cambridge published in 1991. The method is based on measuring the change of attenuation in the blood vessels and tissue following a bolus injection of conventional contrast material. And the technique is now widely used in clinical practice for patients with stroke. When preparing for this lecture, I went back and looked at Lowenthal's book on radioactivity and nuclear radiations, and discovered that the concepts that lay behind CT perfusion imaging were there, but applied within industrial situations. The next major development that led to combined structural and functional imaging was the introduction of hybrid imaging devices such as PET-CT and SPECT-CT, and more recently PET-MRI. And these imaging devices have assigned a number of roles for the anatomical imaging component. We can use the anatomical modality for correcting the nuclear medicine images for attenuation. We can use the anatomical imaging to identify the location of abnormalities identified on nuclear medicine. The anatomical information can provide information about the nature of the lesion and also can detect additional pathology. But in the context of radiomics, the anatomical modality offers a fifth role, which is additional measurements. So, for example, we could use PET-CT combined with perfusion CT to get multimodality imaging of, in this case, lung cancer structure, the glucose metabolism, and using the CT for the blood flow. And we can even derive combined images. This is here the relationship between glucose metabolism and blood flow, an image that was created by a former Lowenthal lecturer, Matthew Griffiths. The third development, which increased the number of imaging measurements that could be made, arose from computer processing methods. And I shall show one example of one that is used for quantifying tumour heterogeneity. So here we have a CT image of a renal cancer, and we can filter this image to derive this information of a tumour. Now, this filter is one that was developed at the University of Sussex for identifying military targets on complex backgrounds, which, in other words, the battlefield. And it was discovered that the computer could discriminate the targets more effectively by filtering the image to highlight coarse features. And it is the same filter, in this case, that has been used to highlight specific features within the renal cancer. We can then look at the range of values within the filtered image in the form of a histogram and derive from that a whole array of histogram descriptors, which then can in turn be used to classify the patient or the tumour into condition A or versus condition B, which may be a pathological state, a tissue type, for example, or it may be a clinical outcome such as survival. And so now, by combining the benefits of hybrid imaging, quantitative imaging, and imaging processing, we're in a situation where from a single examination, we can derive a whole array of image-based measurements, some of them from the anatomical modality, some of them from the PET. And then the next problem is trying to work out how to interpret all this information. And we can borrow the methods used from genomics, and this may be why radiomics has been adopted as a term. So here we have plotted what is known as a heat map, where the colour tells, for each measurement, tells us how far above or below the mean for all of the patients that measurement is, and then we can then cluster the patients accordingly in such a way as we can see here. This group here with condition A tends to have low values for these parameters and higher values for these parameters, for example. And so we can begin to tease out from multiple measurements, tease out different conditions, again, either clinical conditions or pathological conditions. And of course, the latest development in multimodality imaging is PET MRI, which gives us simultaneous molecular and chemical imaging. And PET MRI increases the number of measurements that are available to us even more. And it is worth us spending a few moments to look at some of these measurements, because many people working in nuclear medicine who've not worked with PET MRI may be unfamiliar with some of the concepts. But in many ways, they are similar to measurements made in nuclear medicine. So, for example, most people in the audience would be familiar with the measurement of glomerular filtration rate using chromium-51 EDTA. We inject the tracer into the patient, and then it is excreted over time, and the speed at which the tracer disappears from the blood system is represented by this slope on a log graph, giving us the glomerular filtration rate. Well, exactly the same concept applies to the measurement that can be made on MRI. But here the MRI has labelled the hydrogen atoms within the water molecules within a tissue volume, and these will move out of the volume with time due to diffusion. And so if we plot the signal over time, although very much more rapidly than GFR, this is 1,000 milliseconds, but if we plot the drop-off of signal with time, it tells us how fast the water is moving out. In other words, the diffusion of the water through the tissues. And this is the apparent diffusion coefficient. And then plotting this pixel by pixel, we get a map of ADC, in this case showing restricted diffusion in a prostatic cancer. And in cancer, the diffusion is restricted because the cells are packed tightly together, preventing free motion of the water molecules. Another measurement that is of interest to nuclear medicine people is chemical shift imaging with MRI, because this is the basis of attenuation correction for PET MRI. Here we have an uncorrected image, and this is the corrected image after MR attenuation. Now you may wonder, how could an imaging method that measures water tell us about attenuation of photons? Well, the method is used is chemical shift imaging, where you compare an image where the signal from fat and water are in phase to an image where fat and water signals are out of phase, simply by altering the timing of the measurement. And so here we have an example of a patient in phase image of the upper abdomen, out of phase image, and there's two things I want to highlight. One is that on the out of phase image, the liver has gone dark, and so has the bone marrow. And this is because this patient is a diabetic with fatty liver, and there are approximately equal amounts of fat and water in the liver. And similarly in the bone marrow, as a young patient whose bone marrow has not developed the predominantly fatty composition of older people, and so there's significant dropout in the bone marrow. And so by measuring water and fat, we can characterize tissues as water or fat and assign to those an attenuation value for the 511 keV of protons of the PET. But we can also use this imaging to measure the relative quantity of water within any individual pixel, just by a calculation from these two images. So with PET MR, we can get a whole array of different measurements from the tumour, in this case from a tumour of the larynx. We can use the PET for measuring the glucose metabolism. The ADC, the apparent diffusion coefficient, tells us about cellularity. The change in contrast enhancement after an injection of contrast material tells us about angiogenesis. The water fraction from the attenuation correction image of the PET MRI can tell us about how much water is in a tumour. And then there's a fifth measurement that I haven't described in any detail, which is the so-called R2 star, which is an indication of how much hypoxic blood there is within the tumour. And so with PET MRI, we can end up with a very large number of measurements. We have a large array of primary images. We can then process each one of those, for example, with this texture mapping that I described earlier. From each of those derived images, we can develop a number of statistical maps from the statistical parameters from the histogram, and then we can analyze these in some way to characterize the different groups of patients, either using cluster analysis, as I described before, or using a decision tree where we sequentially apply individual measurements until we can separate out, in this case, wild type and mutant forms of colorectal cancer. So let's summarize then the concept of PET-based radiomics. This is a way of giving us combined diagnostic and predictive molecular imaging. The conventional component of the examination, the clinical FDG PET, gives information that we know is of value for diagnosis. And at the same time, we can get prognostic and predictive information, in which case the PET is used either for localizing tumour sites for analysis, for instance, texture analysis, or we could use the measurements of radiate tracer uptake, and also we could use the measurements from the anatomical modality, anatomical being in inverted commas, because many of these measurements are actually functional. And there are a number of advantages for using radiomics over tissue analysis. The first obvious one is that it's non-invasive. You don't have to stick a needle in to get the information that we're looking for. You also get an image of the whole tissue, not just a little sample, as would be, for example, from a biopsy. We can study multiple disease sites at the same time point, which would require, for tissue analysis, would require multiple biopsies. We can do multiple time points to see how all these measurements change with time. Again, relatively difficult to do with biopsy because of the need for multiple invasive procedures. We also are looking at the phenotype of the tumour or the disease. It's not necessarily oncology, but it could be other diseases. But we're looking at the phenotype rather than the genotype, which is going to be more closely related to the biological behaviour of the disease than the genotype. And because it's done as an adjunct to something we're going to do anyway, the diagnostic imaging, it is a relatively low cost. So it's perhaps unsurprising that the number of papers published on radiomics has increased exponentially in recent years. Now, the other half of the title relates to personalised medicine. So what is that? Well, personalised medicine separates people into different groups such that the medical interventions are tailored to the individual patient. And this is based on the predicted response or the risk of disease. And I shall give two examples of studies that I have been involved in for radiomics. One is a PET-CT based method for treatment selection in advanced melanoma from the Translational Research Institute and the Princess Alexandra Hospital in Brisbane. And the other is a PET MRI radiomics approach, which has potential application for risk-adapted post-operative surveillance in colorectal cancer from University College in London. So here's the concept for the first study. Now, currently, with our current medical approaches, we have a group of patients who are heterogeneous, but they all tend to get the same treatment. Many of these will have a beneficial effect, some will have no effect, and some will have an adverse effect. Now, for radiomics and personalised medicine, the concept is to use the radiomics to select the patients for the treatment that is most beneficial for them. So in this case, the blue patients will still get the blue pill, and the red patients now will have a different treatment which will be effective for them where it was ineffective before, and the patient who had adverse effects previously would have a treatment again which will be beneficial. To understand the potential use of radiomics within advanced melanoma, we need to know something about cancer survival strategies. This is the processes by which a tumour will gain a biological advantage by increased aggression, for example. Now, some tumours achieve this by on account of a gene mutation that switches on the activity of the protein that is encoded. And many of these activating driving mutations lie within a single pathway, the MAPK pathway, which starts off at the cell surface with an endothelial growth factor receptor, passes through a number of molecules, the signal gets to the nucleus and switches on proliferation, angiogenesis, migration and invasion, all of which renders the tumour more aggressive. Now, if we have a mutation that switches on the activity of any of these molecules, we have the potential to treat the tumour with a targeted therapy. But the targeted therapy must be further down the chain than the molecule that is switched on. So there's no point, for example, inhibiting endothelial growth factor if we have a mutation in BRAF. But if BRAF is mutated, we can inhibit BRAF or MEK and achieve a benefit. And this is what happens in many patients with advanced melanoma. The BRAF mutation occurs in approximately 50% of patients with stage 3 and 4 melanoma, and if this mutation is present, we can treat the patient with targeted therapy. But other tumours develop a biological advantage because they have a high number of mutations. Now, this has a number of effects. Some of these mutations will lead to proteins on the cell surface of the tumour, which are antigens, which will switch on an inflammatory infiltrate. And the other thing with a high mutational load is that the tumours will tend to have clonal heterogeneity. And if these clones are sufficiently different in their phenotype, we may be able to pick them up because of differences in their imaging characteristics. And if we have high mutation load, then we know that these patients tend to benefit from immune checkpoint inhibitors, because this can increase the inflammatory response that can help to control the tumour. And this is another treatment option in malignant melanoma, because melanoma is one of the most highly mutated forms of all cancers. You can ask why just not use the genome-based results for personalised medicine. Well, there are a number of reasons why this is limited. Firstly, a significant number of patients have analysis failure due to poor quality DNA. For BRAF, for example, this is somewhere between 4 and 9%. And also, if the tumour of interest is deeply seated, it may be unsafe to biopsy the tumour. And then we do know that even if the gene mutation is present, that a significant number of patients fail to respond to the targeted therapy. And this may be because there is heterogeneity of the mutational status, and the piece of tumour that has been biopsied is not representative of the whole tumour. And there may also be differences between the genotype and the phenotype. Whereas with imaging, we can overcome the technical failure. As I pointed out, we can measure multiple tumours and measure the whole of an individual tumour. And we also we're imaging the phenotype, which can give us information complementary to the genotype. Now, the study that I mentioned before from the Translational Research Institute and the Princess Alexandra Hospital in Brisbane is summarised in this figure. The imaging modality was PET-CT. The CT component went under texture analysis. but the FDG PET information was also used for routine clinical staging, but also and for localization of tumour sites for analysis. The patients also had a range of genomic and scriptomic analysis performed, which I shall show shortly. And then this was validated by using immunofluorescence to show that these genomics was actually translated into changes in protein expression, and there was also a look at survival outcomes in these patients. So the first finding of significance is that the radiomic markers were associated with the BRAF mutational status. And so here we have all of the imaging measurements that were made, and the ones in red are the ones that were significantly associated with progression-free survival or overall survival. And it turned out that that the patients who did not have a BRAF mutation, that the parameter that identified patients without a BRAF mutation was the same one as identified the survival of the patient with over time. The study also showed that the expression of immune-modulated genes is associated with imaging heterogeneity. Validating this concept is that genes that result in an inflammatory infiltrate are also associated with imaging heterogeneity detected with texture analysis. So here, for example, we have the expression of a range of immune-modulated genes, and these have been clustered to identify two sorts of patients, two sorts of tumours: an immune cold tumour, an immune hot tumour, hot being the ones with the inflammatory infiltrate. And we found that the markers that were associated with survival were also detected differences associated with differences in the immune infiltrate. And the same parameters, when combined, was significantly associated with the tissue expression of CD8, which is a protein important for inflammation. Now, the second study uses a slightly different approach for radiomics, and this is selecting patients for more or less intense treatment surveillance. So currently, after surgery for colorectal cancer, for example, all patients are going to treatment surveillance, even though some of them will never develop a recurrence. And these patients potentially could be saved the worry of having surveillance, and also the expense. So we can conceive of having the radiomics used here to identify which patients should have surveillance. And the example that I wanted to show you was the one from University College London, where patients having PET MRI, where the measurements were made of FDG uptake, of apparent diffusion coefficient, and then the water fraction image was used for texture analysis. So there was an array of about 40 measurements that were used to characterize the patients as good or poor prognosis. And it was found that there was a group of patients identified using water texture that had no deaths in a period of 32 months, compared to a higher risk group patients where the deaths occurred. So you could argue that these patients need not undergo surveillance, and you should concentrate the resources of surveillance on the patients who are most likely to end up with recurrence and death. So those two studies are just some examples of many studies which highlight a potentially bright future for radiomics, not only in cancer but in other disease processes. But what are the next steps to achieve this potential future? Well, first, I think we can consider the way in which we find radiomic models that are potentially clinically useful. Now, the traditional approach to medical research is to use a translational model where we go from an early phase of first in humans through to translation of the method into population health. And so in imaging, for radiomics, the first stage would be to identify an imaging correlate for a biological process of known significance, such as an oncogene. We would then do retest measurements and measurements of biomarker prevalence in the relevant population. We would then measurements of biomarker performance, such as hazard ratio and therapeutic impact in a clinical population. And then we could do health economic evaluations to translate that into a population and societal benefit. But with PET-based radiomics, we have the potential to look at this translation pathway in a new way and start at number 3, where we're using them in clinical practice, and or I should say discovering them in clinical practice. And then once we have discovered a potential biomarker, we could do the health economic evaluation next to determine whether it's worth going any further, because if this could not be economically viable, then there's no point doing further research. And then if that was going to be potentially economically viable, we could go back and do these earlier phases. So this is an example of reverse translation. So we can define this reverse translation process as a way of using radiological observations in patient populations to formulate hypotheses that could be tested in the laboratory. And this approach has a number of advantages. Firstly, it is clinically focused and orientated on a specific problem. And secondly, it uses available image data, which has reduced research costs, because we're using methods and tracers, for example, that are already licensed. The translation into clinical practice is fast. And also, we may identify biomarkers that we wouldn't have anticipated from current basic science, and I shall show an example of that. So this is our reverse translational model. We end up going PET-CT or PET-MR. We're collecting our clinical data in the usual way, but at the same time we are exploring for biomarkers that could be associated with a particular outcome. Once we have found those, we can then carry on collecting clinical information in other studies whilst we qualify the radiomic model with test-retest performance, looking for biological correlates and confounding factors, and finally undertake the health economic analysis. Next, I would like to look at how we evaluate radiomic models that we've either found already or will find in the future. And this is an important issue because generally the scientific quality and reporting of radiomic studies has been shown to be insufficient. And this study from 2020 by Park et al. Used a previously developed radiomics quality score to look at the quality of radiomics publications, and this yellow marker is the scores in various domains against what is achievable, and you can see that there is a large shortfall from the ideal reporting quality. And part of the problem is that there is no established way of evaluating radiomic studies, but we can envisage one that is conceptually similar to the way we currently evaluate diagnostic imaging studies. So we would start with technical performance. We would go through prognostic and predictive performance, prognostic and predictive impact, therapeutic impact, and health impact. And I shall go through each of these in a little bit more detail. So the first thing to consider is technical performance. Is the imaging technique reproducible with an identifiable biological correlate? Now, the issue of reproducibility is really one of quality assurance. We need to know that the method is effective regardless of the imaging platform used. We need to know whether the methods or the results are sensitive to changes in the image acquisition and image processing. Commercial software would be helpful in this regard because it would encourage conformity across different platforms and different sites. And there is also an issue of regulatory approval, which could actually be quite difficult for a biomarker as opposed to an imaging device. Now, the issue of biological correlate is also important because the biological rationale for a medical decision making will be important for clinical acceptance. If we have a method that effectively identifies patients with a particular condition or a particular prognosis, but we can't explain why it has characterised the patients in that way, a so-called black box, then these methods are unlikely to be acceptable to clinicians. The other issue we need to consider is around the relationship between the imaging phenotype and the genomics. Now, it's possible that if we have a known genomic or biological characteristic that is already identified to be clinically significant, if we focus our radiomic development for correlates of those, that may speed up the radiomic development, but it also it may not turn out to be that simple. Sometimes the relationship between the imaging phenotype and the genotype can be complex, and I'll just show an example of that. And also sometimes the biological correlates that we find may not be prognostic themselves, so what do we do then? And I shall show an example of that. So in terms of the differences between genotype and phenotype, I'll go back to the MAPK pathway, but this time in colorectal cancer, and a particular study that looked at FDG PET, texture analysis, and perfusion in rectal cancer. And the intention was to identify mutations in the KRAS gene, which are relatively common in colorectal cancer. And it was found that there were two phenotypes for the same gene mutation, for the KRAS mutation. And using this recursive decision tree method I alluded to earlier, some of the patients with the KRAS mutation had high glucose metabolism, that's an SUV max above 16, and those also had a particular texture characteristic. But there are also a number of patients, approximately a third of the total mutations, that had a low SUV, and these were identifiable on the basis of the same texture parameter and blood flow. So we have a high metabolic phenotype and a low metabolic phenotype which has high blood flow for the same genotype. And it is entirely possible, I don't know in this particular case, but it's entirely possible that these two phenotypes from the same gene mutation would benefit from different treatments. Now, I also mentioned that sometimes the imaging correlates do not turn out to be prognostic themselves, and this is some work from Princess Alexandra Hospital, where the biomarker that was looked at was simply measuring the number of pixels within the tumour. The tumour was identified by from a PET region of interest, but the measurement was simply the number of pixels with a density of less than 20 Hounsfield units. And in non-small cell lung cancer, this was shown to be significantly prognostic. Now, in a public available data set of lung cancer images, also had their genomics to go with them, it was possible to identify 30 gene correlates, which was significantly different between patients with high values of high number of pixels above 20 Hounsfield units from those with low. And then with cluster analysis, we're able to show that these genes were definitely related to the CT measurement. So this is the darker the grey, the higher the proportion of pixels with of less than 20 Hounsfield units. You can see that it's clustered quite nicely. These genes have low expression when there's a high value for the biomarker, and these ones have low expression of these genes when this biomarker is less likely to be present. But then, when looking at the known studies on whether these genes were prognostic, it was found that none of these genes were prognostic. So here we have an imaging marker that is prognostic, whereas gene correlates are not. But we did find that a number of these genes were related to keratin, and keratin, that we know, is of high density on CT. So there is a biological reason for the CT measurement being different in tumours, but there's not a reason for it being prognostic. And this is presumably related to epigenetic factors that alter the protein expression of the individual genes. So the next stage in the evaluation is to look for prognostic and predictive performance. And this is where most studies have been performed to date. And does the imaging technique accurately determine prognosis or predict treatment response? But it's important to do this in the right way, using consistent evaluation criteria and reporting guidelines to minimize bias and avoid overly optimistic conclusions. And another thing that is often missed from these studies is the we need to have a comparator in the study. That could be an established clinical or pathological marker of prognosis, for example, or even simpler imaging measurements such as tumour size. So a complex radiomic model is unlikely to be useful if it produces no more information than a simple measurement of the size of the tumour. So the key factors for reporting prognostic performance are the hazard ratio. This is the difference in survival functions of two populations. That could be overall survival or disease-free survival. So here we have the proportion of patients surviving over time. In this particular case, if you're in the red group, you are twice as likely to have to die in the follow-up period than the green group. But just measuring that isn't enough. We need to do multivariate analysis or potentially a randomized control trial, although this is not essential. But we need to show that the measurement is independent of stage and treatment and other known prognostic factors. So the classical way of ensuring an unbiased estimate of a threshold that which distinguish patients on the grounds of their survival, so an imaging measurement threshold, is to use to develop the threshold or the radiomic model in one cohort of patients, and then test it on a separate cohort of patients. And then we avoid any bias of using the same cohort for identifying the threshold and testing it. That can actually take a lot of patients. So there are alternative methods where we divide the cohort up. In this case, it's 2, but it could be 5 or as many as you like, and we use one group for developing a threshold that we actually test on the other group. And this so-called cross-validation is another way of avoiding bias without having to have a very large number of patients. So what about prognostic and predictive impact? This asks the question, does the imaging technique change prognostic or predictive confidence or the use of other prognostic or predictive markers? And there are very few studies that have actually looked at this for radiomics, and yet it is potentially relatively simple to do. And this is an example I've used of a hypothetical radiomic model with a hazard ratio of 2 and a prevalence of 50% for patients with non-small cell lung cancer. And I've modelled this using a publicly available software adjuvant online, which has a factor where you can put in biomarkers. And so, for example, in this particular case, we can see that the 5-year survival for a patient, for all patients without chemotherapy is 58.4%, with chemotherapy is 64.2%. So the survival benefit from chemotherapy is 5.8%. But if we deploy the biomarker, we can see what those figures are for patients in the good prognostic group and in the poor prognostic group. And we can see that if you've got a poor prognosis, your survival benefit from the chemotherapy is greater than it would be if you had in the good prognostic group. And this could potentially make a difference to the treatment, which is the next phase of the assessment, the therapeutic impact. And again, this is just modelling the same information, but here with a plot of pre-test 5-year survival improvement from chemotherapy versus post-test. So the blue line is the line of identity. This is without deployment of the biomarker. And it is generally considered amongst lung cancer clinicians that for platinum-based chemotherapy to be worthwhile in patients with non-small cell lung cancer, you need to have a 5% improvement in 5-year mortality. Otherwise, the downsides of the chemotherapy don't justify the benefit. So the green line is the good prognostic group. Following, it shows you that if you're, for example, here, if your 5-year pre-test survival is 4% after deployment of the bar. If you're in the good prognostic group, it's 3%. If you're in the poor prognostic group, it is just about 5%. So there is very little difference in deploying the biomarker. The therapeutic impact is zero, because in either case the patient wouldn't be considered to benefit sufficient from chemotherapy. But these shaded areas here represent the patients conditions where a patient would change their treatment based on deployment of the biomarker. So here, for example, if you've got a pre-test survival probability benefit from chemotherapy of 5.5%, this reduces to just over 4% in the good prognostic group, so you wouldn't benefit from chemotherapy. Whereas on this side of the curve, if you're at 4.5%, you wouldn't consider generally to be worthwhile having chemotherapy, but you would if you fell into the poor prognostic group, shaded in blue here. So this is a way of looking at how often the patients would the deployment of the biomarker would change the treatment. And then the final question: health impact. Does the imaging technique lead to an improvement in patient survival or morbidity? Currently, there are no studies I've been able to find where this has been done for radiomics. And again, we could just use a modelling technique where we compare the quality adjusted life years from standard treatment to groups separated by the biomarker. It's simply by plugging in the hazard ratio and the biomarker prevalence. Now, the other aspect of the health impact is whether that health impact is worth the extra cost. And there are some potential health economic impacts of using radiomics. And these really follow on from what I've shown already, that if you use radiomics to target post-treatment surveillance to high-risk patients, you will save the costs of by some patients not needing to undergo surveillance. And for treatment selection, we could actually avoid we could save the costs of treatment that is unbeneficial if the biomarker is present. And again, by using modelling, it's possible to show that to use PET MRI radiomics in this way has the potential to convert PET from being cost ineffective to being cost effective. Currently it is estimated that for PET MRI to be used for diagnosis alone, it is unlikely to be cost effective unless incremental cost over PET CT is less than $2,000. But by modelling, we can show that if we use PET MRI for treatment selection or image guided surveillance, that we could have situations where PET MRI would be cost effective even with an incremental cost over PET CT of $4,000. So PET MRI perhaps could be this could be one use for radiomics could be one way of moving PET MRI into a clinically cost-effective technique. Now, the theme of this meeting is bridges to new horizons, and so I thought I'd finish off with this, considering how we can use a bridge from this 30-year-old view of nuclear medicine and radiology as separate to one where we can combine the two by use of radiomics for guiding treatment. And I've summarised this with a picture of Brisbane's Story Bridge. And here we have a group of tourists who have decided to walk the bridge. And this is where we might be now on the road to radiomics in clinical practice. We've walked up the steep side, developed biomarkers, shown them to be potentially useful, and we're now on a downward stretch. But we can see before us all the difficulties required before we can get these techniques into clinical practice. Now, either we can do like the visitors to the story bridge, cross over and go back up on the other side and go back to where we started, or we can push on and do what's necessary to get these techniques into clinical practice. And I think there is a role for the Society of Nuclear Medicine here. Now, the Society of Nuclear Medicine has an important role in education, and this is going to be a key component for moving radiomics into clinical practice. We need to train nuclear medicine specialists and technical staff, and even researchers and those in academia are sometimes unaware of the issues required to get their techniques into clinical practice. The Society has an interesting qualities and standards, and as I've pointed out, these are going to be important for getting radiomics into clinical practice. We're going to need software for extraction and reporting and delivery of radiomic data, and we need to have standards for consistency across imaging platforms. And the Society also aims to have close ties with stakeholders, and this is going to be important if we're going to convince clinicians that this is a technique that is worthwhile of consideration. But if we could achieve all these things, then we could envisage a future where radiomics is used to guide patients' treatment and their imaging surveillance. Thank you.